Hi, my name is Dave Gordon. I'm a bioinformatics software developer in the Institute for Genomic Medicine at Nationwide Children's Hospital, and my talk is about the work we've been doing to use machine learning to help guide treatment of cardiac patients with genomic variants in lamin AC. I'd like to start off by telling you about some super cool cutting-edge methodologies I've invented to completely solve this problem. However, I'm not going to because, yeah, well, honestly, that feels good to get out of the way early. Sorry to disappoint any of you who expected more, but, well, wait, maybe I'm getting ahead of myself. Let's take a step back for a moment. What is lamin AC? Well, it's a set of proteins that are translated from the LMNA gene found on the Q arm of chromosome 1. Through alternative splicing, the gene produces various A-type lamins that are important to signaling, transcription, and the organization of chromatin in the cell nucleus. The protein has three main regions: an N-terminal head, a helical rod, and a C-terminal tail, and the proteins dimerize through the helical rods wrapping around each other. Problems with the protein can cause a spectrum of diseases termed laminopathies, but today we focus on cardiac laminopathy, which can manifest as phenotypes such as arrhythmias, atrioventricular block, and dilated cardiomyopathy. These are usually associated with missense or nonsense variants in the head and rod regions of the protein. Our project focuses on the tougher to evaluate missense variants. A patient was referred to our cardiology department with a personal history of atrial fibrillation and complete heart block, which is to say the electrical signals are unable to pass from the atria to the ventricles in the heart. They came from a family with a 4-generation history of unexplained sudden death and various arrhythmia-related phenotypes. Per the American College of Medical Genetics guidelines, a commercial sequencing lab returned a variant in the LMNA gene termed a VUS, or variant of unknown significance. For those who aren't familiar with these guidelines, this is essentially the clinical equivalent of the shrug emoji. It's better than overstepping the bounds of what we can be medically sure of when treating a patient, but as far as helping direct care, it's really not great. With familial consent, we began researching the variant, a fairly conservative replacement of aspartic acid with glutamic acid at position 136, or D136E. We performed functional validation on cell lines from both affected and unaffected patients from the family. Immunofluorescence and Western blotting showed a decrease in the expression of lamin AC protein in cell lines from affected patients, and QRT-PCR in cell lines from affected patients showed decreased mRNA levels. While the functional studies showed that the variant had some effect, we wanted to make sure that this was actually causative for the disease we saw in this family. We performed Sanger sequencing of the D136E LMNA variant among 4 affected and 3 unaffected individuals, and found that it did segregate, only appearing in the affected individuals. We also wanted to rule out other genes, so we performed exome sequencing of 3 affected and 1 unaffected family member to eliminate other possibilities. The only other variant of interest we found was in the gene Titin, or TTN, but we found that it was not consistent with the phenotypes we saw, and in ClinVar it had been previously reported as likely benign in some cases. Adding the co-segregation of the LMNA variant to the absence of the variant in NOMAD, the low rate of benign mutation and high conservation in the region, and the established disease mechanism in LMNA, we were able to reclassify this variant as likely pathogenic per the ISMB guidelines. So, with apologies to Ali Brosh of hyperbole and a half, this leads us to the obvious conclusion: we need to sequence all the things. But, of course, this isn't wholly pragmatic. The cost might be overcome, but also the availability of the family, consent issues, adopted children, etc. are hurdles we can't necessarily overcome. So this is a translational medicine talk. How do we take what we know and translate it? Well, we add software, which from history we know solves everything? We hypothesized that we could use machine learning over the spectrum of known LMNA missense variation to classify variants as either pathogenic or benign. Easy enough. We planned to train a classifier by using the spectrum of known missense variation in LMNA, labeling variants as benign that are either in ClinVar as benign or likely benign, or in NOMAD at a frequency that we can presume is benign based on the disease prevalence. We'd label variants as pathogenic that are in ClinVar previously reported as either pathogenic or likely pathogenic. we'll use the majority of the variance to teach the model to recognize the differences in both computational and biological features between the benign and pathogenic variants, and we'll also reserve a validation set so that we can just confirm that our training did a good job. Now, this was all well and good until we tallied up our variants and realized that there was one benign variant. One. Now, it is really hard to train a classifier when one of your categories is basically non-existent. So that didn't work at all. This is where you can picture me, the failure from slide 2, sitting sadly, staring out a window in the rain. Well, we've still got about 5 minutes here to translate some medicine, so let's regroup and examine what we do have. We have a set of variants, some pathogenic, mostly unknown significance. Can we leverage existing bioinformatics scores? Well, they're not perfect, but they should give us a better chance than a random guess. Think of it as a British accent in Star Wars. With the notable exception of Obi-Wan Kenobi, it's a pretty good indication you're not up to any good. And seeing as how ISMB is squarely in Big Ten country this year, we can also apply that to other domains. That said, having a British accent and making an unfortunate choice of universities, like computational variant scores, aren't perfect predictors. Please do go check out these talks by two of the nicest and brightest scientists I have the privilege to work with. So we have these predictive scores. We know they're not perfect. They're trained across the whole genome or exome, not specific to LMNA. We have a set of ClinVar variants reported as pathogenic or likely pathogenic. It's probably not perfect, but it's probably a pretty good label. And then we have many variants that have been previously observed in Nomad, we'll call it a quote control population. It's likely imperfect. In this case, conduction defects can develop over time. Nomad is presumed healthy individuals, but if they were sequenced before something was diagnosed, they would still be in there. Our new hypothesis is this. While we can't train a classifier, we can cluster variants into similarly scored groups and stratify their risk of pathogenicity. We'll mitigate imperfect scoring algorithms by projecting an ensemble of variant scores into 2D space using UMAP. We'll cluster them using k-means, and then we'll use chi-squared tests to determine if there is a significant association between our clusters and rates of reported pathogenicity. And here are the results. We used the elbow method to determine that 3 would be an appropriate k-value for clustering. While I'd love for the clusters to be more discrete, the smallish number of variants and the continuous scores used as features leave us with a tighter plot. We then look to ClinVar to see if our clustering gives us any useful stratification of pathogenicity risk. And indeed, we see that clusters 1, 2, and 3 have significantly different pathogenicity rates of 38%, 15%, and 6% respectively. We also see that our functionally validated variant, D136E, falls in cluster 2. As mentioned earlier, ClinVar itself might be an imperfect measure, so we dig a little deeper. This plot is a little busy, but when you look at the red dots denoting 2-star and greater variants, where ClinVar has multiple non-conflicting reports of pathogenicity, we see an even more dramatic trend with no variants at all in cluster 3. While we're very pleased with the results of our study, there are some limitations we need to acknowledge. There's possible overlap in the methodologies between the computational predictions we used as features. There's the lack of a solid control set, given the fact that undiagnosed subjects could possibly be included in Nomad. There is probable publication bias in ClinVar, where benign variants are not reported at the same rate as pathogenic variants. And there could even be the possible bias of ClinVar data if some labs reported variants because of the same predictive scores that we were using in our study. I'd also like to highlight a few future directions for this work. We'd hypothesized that the functional validation of variants in clusters 1 and 2 may have a high positive yield, and that knowledge would feed back to better models in the future. We see the addition of new and improved predictive scores and biological machine learning features as improving our model. We'd like to apply this methodology to other genes and regions of the genome with limited benign variation to see if it could be useful there. And we want to begin using this clustering methodology to augment clinical care by stratifying the risk for patients found with missense LMNA variants. While I hope this work is interesting and helpful to you, I'd like to leave one more takeaway. In science, we are all failures at some point or another, but often we can find ways to pick up those pieces and reform them in a way that still translates to our shared mission of better outcomes for our patients. I'd like to thank Dr. Jeff Bennett, my co-first author on the paper in Heart Rhythm, and the one who came up with the idea of using machine learning in this domain, to Drs. Kim McBride, Peter White, and Vidhu Garg, senior authors on our paper, to our other co-authors, and to all our friends and colleagues at Nationwide Children's Hospital, and finally to you for your attention today. Thank you very much.